Hello students, welcome to EPG Pathshala. I am Dr. Hari Prasad, faculty at the Department of Biophysics, All India Institute of Medical Sciences, New Delhi. Today I am going to talk to you about non-gel based proteomics from the paper Techniques in Molecular Biophysics Part 1. The learning objectives in this module would be Introduction, Necessity of non-gel based proteomics, Classification of non-gel based proteomics, Isotope labeling using ITRAC, ICAT and TMT, metabolic labeling that includes CLAC, nitrogen labeling, proteolytic 18 oxygen labeling, absolute quantification, QConcat and PCPS, label-free proteomics that is MUDPIT, data dependent acquisition that includes spectral counting and spectral peptide intensity, isotopic labeling or label-free approaches, advantages and disadvantages of gel-free proteomics. Before we get into this particular module, let's try to understand the concept of proteomics. A proteome is a protein-material of a protein and genome. This was conceived by Professor Mark Wilkins in the year 1994. The proteome is the entire set of proteins produced by an organism or a particular tissue in the human body at a particular point of time. Proteomic profiling is an experimental platform to map all the proteins expressed in a given phenotype, and the differential expression compares the protein profile in two or more phenotypic states. When we talk about clinical proteomics, it includes the proteomic analysis of human body tissues. Towards this, there are two effective techniques. They are gel-based proteomics and non-gel-based proteomics. Before we get into this particular module, that is non-gel-based proteomics, let's just try to see the different disadvantages that rest with gel-based proteomics. In the gel-based proteomics, quantitative reproducibility is very difficult. There is poor representation of low abundant proteins because they are masked by all the high abundant proteins. There is difficulty in profiling highly acidic or highly basic proteins. Difficulty also lies in profiling proteins with extreme size or hydrophobicity. There are difficulties pertaining to automation of the gel-based techniques. Co-migration of proteins in a single spot makes quantification inaccurate. Interference of non-protein substances affect gel resolution. And finally, this particular concept of gel-based proteomics is not suitable for large number of samples. Classification of methods in gel-free proteomics. First, we have the label-based proteomics. Under this, we have chemical in vitro isotope labeling, which comprises of isobaric tags for relative and absolute quantification. ICAT has isotope coded affinity tags, and TMT, tandem mass tag. Then we have metabolic in vivo isotope labeling, which comprises of CLAC, that is stable isotope labeling with amino acids in cell culture, the N1415 labeling, oxygen 1618 proteolytic labeling. We also have absolute quantification by QConcat and peptide concatenated standards. Under label-free gel-free proteomics, we have MUDPIT, data dependent acquisition and data independent acquisition. Gel-free proteomics. The question is to label or not to label. Labeling proteomic methods are based on the fact that both labeled and unlabeled peptides exhibit the same chromatographic and ionization properties but can be distinguished from each other
by mass shift signatures. Label-free proteomic quantification is based on the fact that peak intensity of peptide ion is proportional to the ion signals during the mass spectrometric analysis.
Now we come to the concept of eye track. Eye track refers to isobaric tags for relative and absolute quantification. It comprises of an amine-specific reactive group. This tags to the primary amine group of lysines. Reporter group has different isotopic composition with 1 Dalton mass shift. There is also a balance group that has an isotopic composition complementary to the reporter group. The sum of reporter and the balance groups is always a constant irrespective of the different versions of the tag and comes up to 145 kilo Dalton. Tagged peptides are isobaric and are therefore not discriminated in mass spectrometry. Fragmentation in MS-MS releases the reporter ion during collision-induced dissociation. Relative abundance of the peptide is deduced from the relative abundance of the corresponding reporter ions displaying distinct masses from 114 to 117 kilo Dalton. Now, what are the salient features of eye track labeling? It can multiplex several samples and generate quantification data. The data outcomes have increased analytical precision and accuracy. It can compare 4 or more samples in just one experiment. It is useful for quantification of proteins from different samples. Example, when you want to compare the protein profiles of different stages of a particular disease. You want to compare the treatment outcomes across different patient groups. Biomarker profiling, replicate measurements of same sample. It is also a patented technology from GE Healthcare. Eye track labels fragment to produce reporter ions that enable relative quantification in a silent region, usually the unpopulated at low mass to charge range of 114 to 121 Dalton.
We shall now discuss some of the applications of an eye track in clinical proteomics. In our lab, we did the proteome analysis of the macroscopically affected colonic mucosa of Crohn's disease and intestinal tuberculosis. The differentiation of these two diseases is very important in terms of treatment and prognosis. So we have 4 tissue samples comprising of Crohn's disease, we have intestinal tuberculosis, the healthy control, and finally the internal standard. The 4 tissue samples are used for proteomic extraction, and these proteins are subjected to enzymatic digestion using trypsin. The digested peptide fragments from these 4 samples are labeled with eye track reagents. The labeled samples are then mixed and then fractionated using cation exchanger. This particular sample is then subjected to mass spectrometric analysis. During the liquid chromatography based MS-MS analysis, there is identification of the proteins by using the peptide sequence, and the quantification of the peptides are got from the fragmented and released reporter ions. This helps in identifying potential biomarkers which are differentially expressed in the two conditions of Crohn's disease and intestinal tuberculosis. So, in continuation with the eye track experiment, we identified trefoil factor 3 as a potential biomarker to differentiate Crohn's disease and intestinal tuberculosis. As you can see in the table on the left, the protein has more than 10-fold difference across the two phenotypic conditions. The expression of this particular protein trefoil factor 3 has been validated using immunohistochemistry, and as can be seen from the slides on the right, there is an increased expression in the intestinal tuberculosis as compared to the Crohn's disease. ICAT, or isotope coded affinity tag, which is a non-isobaric in vitro labeling technique. In this particular technique, thiol-specific reactive groups for cysteine tagging are used. Biotin moiety-based affinity enrichment of tagged peptides take place. The linker group between them contains 8 hydrogens in the light tag and 8 deuteriums in the heavy tag, which introduces a mass shift of 8 Dalton, which will help in differentiating and comparing. The thiol-specific reactive group for cysteine tagging is 26.6% peptides and comprises of 96% of the human proteome. This particular thing sometimes is overcome by what you call as the isotope coded protein labeling. Yeah. TMT, or tandem mass tag. This is a MSMS based quantitative method that uses isotopomer labels. It employs N-hydroxy succimide chemistry that tags primary amine groups. This allows multiplexing of several samples by chemical derivatization with different isoforms of same isobaric tag that appear as a single peak in mass spectrometric scan. The release of the daughter ions in MSMS analysis, which could range between 126 to 130 Dalton, is used for relative quantification. This TMT reagents enable selective labeling and reactive quantification of cysteines containing peptides from up to 6 biological samples. TMT and ITRAC are similar in terms of quantitative precision and accuracy. However, the number of identified peptides or proteins is higher with the ITRAC as compared to a TMT 6-plex. Metabolic labeling (CLAC), which stands for Stable Isotope Labeling in Cell Culture. This is a simple and straightforward approach for in vivo incorporation of labels into the proteins for mass spectrometric based quantitative proteomics. This relies on the metabolic incorporation of what you call as the light or the heavy form of amino acid by incorporation of amino acids with substituted stable isotopic nuclei, which could be from deuterium, carbon, or nitrogen. Two cell populations are identical except that one of them contains a light and the other contains a heavy form of a particular labeled L-lysine. It is efficient and reproducible as the incorporation of the isotope label is 100%. It works perfectly well for auxotrophically cultivable organisms. CLAC labeling and its variations. There are three types of CLAC labeling. They are standard CLAC, pulsed CLAC, and dynamic CLAC. In the standard CLAC, two populations of cells are cultivated in cell culture. One of the population is fed with growth medium containing normal amino acids, while the second is fed with growth medium containing amino acids labeled with heavy isotopes. The ratio of peak intensities in the mass spectrometry for such peptide pairs reflect the abundance ratio for the two proteins. Next, the p-CLAC, or the pulsed CLAC, is a variation of the CLAC method where labeled amino acids are added to growth medium for only a short period of time. This allows monitoring of the efficiencies in de novo protein production rather than the raw concentration. Finally, we have the dynamic CLAC. As we all know, the proteome of a system is very dynamic with the continuous protein synthesis and degradation. Here, labeling and monitoring of dynamically changing proteome is observed in cellular organelles, thereby which are involved in cell growth and differentiation. Metabolic labeling using nitrogen 14 and 15 labeling. Label is introduced to whole cell or organism through growth medium containing 15 nitrogen labeled inorganic salts, typically potassium nitrate. Quantification process is based on the intensity of extracted ion chromatograms of survey scans containing the pair of labeled, that is the nitrogen 15, and the heavy and the unlabeled, that is the nitrogen 14 light peptide isoforms. Amino acids will incorporate the label, thus mass shift will be peptide sequence dependent. The advantages are the isotopic tags are incorporated into the early stages of the sample preparation and thus reduce variation between samples, yielding highly accurate quantification, and also this allows large-scale quantitative analysis. Proteolytic oxygen 1618 labeling. This particular method is simple, fast, and reliable method that takes place during enzymatic proteolytic digestion in the presence of water with 16 oxygen isotope and the labeled water which has the isotope oxygen 18. Catalytic activity of serine protease can exchange both C-terminal oxygen atoms with the heavy oxygen from water in surrounding solutions. First, the oxygen 18 is introduced upon cleavage of peptide amide bond, while the second is introduced when peptide is bound to enzyme as an intermediate. Resulting peptides, which could be 2 or 4 Dalton heavier than unlabeled counterparts, are pooled with unlabeled peptide mixture and peak intensities of isotopic envelopes and compared and resolved on mass spectrometer. The drawback in this particular technique is that the exchange reaction is rarely complete for all peptides, resulting in complex isotopic pattern due to overlap of unlabeled, single, and double labels. Absolute quantification using QCon-CAT. In this strategy, peptide concatenated standards or tryptic peptides to be quantified are selected from the target proteins and mixed to generate an artificial protein, which serves as a stable isotope labeled standard. The QCon-CAT proteins can be labeled by nitrogen 15 using ammonium chloride as a sole nitrogen source in the medium to specific labeling with either carbon lysine or the carbon arginine, which ensure that for a tryptic proteolytic peptides, each has a constant mass offset of 6 Dalton. Peptide concatenation not only guarantees equimolar spiking of every standard peptide, but also eliminates the need to prepare many standard molecules, as each peptide included in the QCon-CAT is the tryptic peptide per se. Absolute quantification using peptide concatenated standards. In this technique, tryptic peptides in the peptide concatenated standards carries its natural flanking sequences at both sides to faithfully recapitulate the efficiency of proteolytic cleavage in the target protein. Even a peptide excised less efficiently from the target protein, which is colored in pink, can be precisely quantified with the PCS because its standard, which is colored or shown in red, is also excised from the peptide concatenated standards at a similar efficiency.
Disadvantages of labeling based quantitative proteomics. It requires expensive labeling reagents. It requires high amount of starting samples. Multiple sample preparation steps result in a considerable sample loss and reduced detection sensitivity. There is what you call as a ion suppression effect. Mass spectrometer based reduced detector response or signal to noise ratio as a manifested effect of competition for ionization efficiency in the ionization source, that is between the analyte of interest and other endogenous or exogenous molecules. Example, plasticizers extracted from the plastic tubes and the mobile phase molecules that have been added, and the species which have not been removed from the sample matrix during sample preparation.
MUDPIT or multidimensional protein identification technology. MUDPIT is used especially for the analysis of complex protein mixtures. This particular technique involves protein samples which are subjected to enzymatic digestion with trypsin, and the resultant peptide mixtures are so separated by a strong cation exchange and reverse phase high performance liquid chromatography, and are subsequently subjected to mass spectrometric analysis. They have a lot of advantages in that they are fast, sensitive, they have good reproducibility, and generates an exhaustive list of proteins. These could be applied for large-scale cataloging of proteins in organisms, profiling of membrane and organelle proteins, identification of protein complexes, and determination of post-translational modifications and quantitative analysis of protein expression.
MSMS based label free, gel free methods. First of all, the principle: peak intensity of any ion should be proportional to its abundance, which is the ion signals in mass spectrometric analysis. Recent advances in LCMS approaches has allowed correlation between protein abundance and the peak areas. That is, the number of MSMS spectra has widened the choice of analytical procedure in quantitative proteomics. In the technique, for two samples that need to be compared quantitatively, liquid chromatography associated MSMS experiment is performed for both the samples separately, and precursor ions, mass to charge, and the retention time RT file for all the MSMS spectra of each identified protein is created. Depending on the mass spectrometric acquisition mode, two analytical methods, that is data dependent analysis and data independent analysis, could be carried out. The MSMS based label free methods, data dependent acquisition, are also called the DDA. In this method, acquisition of MS survey scan for an allotted period of time by precursor ion selection based on its intensity for subsequent fragmentation. The quantification relies on the assumption that in data dependent acquisition analysis, the sampling probability of a protein is a function of the protein's abundance in the sample. Quantification is done using either spectral counting or spectral peak intensities.
Spectral counting is another label-free technique for quantification. It involves peptides from abundant proteins that will be fragmented, thereby producing higher number of MSMS spectra. It largely depends on the number of spectra obtained and the coverage of sampling. The relative difference is estimated by protein abundance index, or also called as the PAI, which is the ratio of experimental peptides to the number of theoretical peptides for each protein within the given mass range of employed mass spectrometer. Exponentially modified protein abundance index, which is equal to 10 raised to the power of PAI minus 1, takes into fact that generally more peptides are detected for larger proteins and is directly proportional to the protein content in the sample. APEX, or the absolute protein expression, measures the absolute protein concentration per cell from the proportionality between the protein abundance and the number of peptides observed by using a correction factor that correlates the likelihood of peptides observed to their intrinsic characteristics, which could be the length or the amino acid composition. Spectral peak intensity is another label-free technique for quantification. Relative abundance of the same peptide in different samples can be estimated by the precursor ion signal intensity across consecutive LC-MS runs, given that measurements are performed under identical conditions. It is applied to quantitative profiling of purified proteins to identify bona fide components of a protein complex and reveal dynamics of protein-protein interactions. The technique involves that peptide signals are detected, which is in turn reflects the patterns tracked across the retention time dimension, which further talks about the reconstruction chromatographic elution profile of monoisotropic peptide mass, which gives the quantitative measurement of the original peptide concentration. Systemic errors in liquid chromatography and mass spectrometric analysis is normalized by spiking an identical amount of standard protein into every sample to be compared and using average intensity ratios between target peptide and co-eluting peptides as a pseudo internal standard. Data independent analysis. This involves a quantitative comparison of ions emanating from identically prepared control and experimental samples is done using a reproducible chromatographic separation system along with high mass resolution and mass accuracy of an orthogonal time of flight mass spectrometer. The instrument alternates between low collision energies, which is for accurate precursor ion masses, and high collision energies, which is for the peptide fragmentation data. Multiplex parallel fragmentation data yields uniform product ion information of all peptides across the entire chromatographic peaks, which provides continuous mass spectrometric data throughout the entire acquisition. Product ions are time aligned and correlated to precursor ions to generate a list of exact mass retention time, or also called as the EMRT, which are integrated and compared to determine differences in the relative and absolute protein quantification.
Advantages and disadvantages of MS based label free methods. First, the advantages. It helps to quantitatively compare all the peptide constituents in that particular sample. Disadvantages. Replicate dependent makes it very difficult. The susceptibility to errors due to parallel sample processing and analytical variability. It becomes mandatory to carry out sample preparation and MS data acquisition cautiously. You come onto a particular problem called as the matrix effect. This is the chromatographic separation between the two runs need to be controlled to quantify the MSMS identified peptides. This particular workflow is applicable only to high resolution mass spectrometry based platforms.
Isotopic labeling or label-free approaches. First, we will discuss isotopic labeling. Under isotopic labeling, samples can be mixed very early in the workflow, thereby avoiding biases. It is amendable to high accuracy studies using first generation QTOF devices or quadrupole ion trap machines. The comparison of multiple samples are possible, where the platforms of ICAT, TMT, and iTrack are used. Next, we come to MS-based label-free technologies. This requires nano HPLC separation and latest generation mass spectrometers. Sample preparation and data acquisition are two steps that is reproducible. Assuming a convenient correction factor, absolute quantitative data can be procured. Data accuracy could be a drawback, does not detect fold changes lower than 2. Only proteins with major differential expression, which could be potential biomarkers, are detected.
Advantages and disadvantages of gel-free proteomics. First, the advantages. It has higher sensitivity. It is easier automation of procedures that are involved. It has better reproducibility. It has reduced influence of intrinsic protein parameters. There is availability of sophisticated mass spectrometry machines these days. Quantitative data is statistically more robust. Now, what are the disadvantages? The detection of protein isoforms can sometimes be a problem. The intrinsic limitation of mass spectrometry based peptide analysis, or what we have previously discussed as the ion suppression effect.
Validation proteomics or selected reaction monitoring. SRM or selected reaction monitoring is a non-scanning mass spectrometric technique performed on triple quadrupole like instruments and in which collision-induced dissociation is used as a means to increase selectivity. In SRM experiments, two mass analyzers are used as static mass filters to monitor a particular fragment ion of a selected precursor ion. The specific pair of mass to charge values associated to the precursor and the fragment ions selected is referred to as the transition and can be written as the parent mass to Z as compared to the fragment mass to Z. Unlike common mass spectrometric based proteomics, no mass spectra are recorded in a SRM analysis. Instead, the detector acts as a counting device for the ions matching the selected transition, thereby returning an intensity value over time.
Validation proteomics using multiple reaction monitoring, or what we usually call as the MRM. MRM, or the multiple single reaction monitoring transitions, can be measured within the sample experiment on the chromatographic time scale by rapidly toggling between the different precursor and the fragment pairs. Typically, the triple quadrupole instrument cycles through a series of transitions and records the signal of each transition as a function of the elution time. The method allows for additional selectivity by monitoring the chromatographic coelution of multiple transitions for a given analyte. Although broadly used, the term multiple reaction monitoring to indicate the parallel acquisition of multiple SRM transitions might be used in the near future. Over the last few decades, there have been a lot of expressive landmark events in proteomics. Ever since the STS page that was first introduced by Lamely in the year 1970, there have been a lot of flagship research innovations in the field of proteomics. The entire list of these innovative techniques and procedures is listed in this particular slide for the use and reference and study by the students. So students, let us now summarize what we have learnt in this particular module. Proteomics is a promising platform to complement genomics to understand the molecular events that dictate pathophysiological process in the human body. Gel-based proteomics have a number of pitfalls, making it difficult to rely upon in clinical proteomics or biomarker discovery. There are a number of label-based and non-label-based non-gel proteomics approaches. Each technique has its own advantage and disadvantages and is used based on the need, the sample size, the instrument, and the expertise available. Both relative and absolute quantification is possible using non-gel based proteomics. Non-gel based proteomics holds plenty of scope in adding significant data to both basic as well as applied fields of biology and medicine. Thank you so much.